am very involved in genetics and the dairy industry, and I'm also chair of the Alberta Livestock and Meat Agency, which is a sponsor here. So I'd like to invite up all of our speakers. The room might be half empty if we fill all these seats, but you all know where you're going to sit in order. Graham, you start here, and then we have our salmon specialist, and away we go here. Good. Now we will go through the official panel and then get on to things. I do want to say that it's indeed a pleasure to be here, and I want to thank Pierre, and special thanks the amount of work that Cindy and Corinne put in just to this small segment. I can't imagine amplifying that through all of the activities at this conference. So I'm very impressed with the organization here and truly honored to be able to moderate this session. We have a saying at the Calgary Stampede, actually, because agriculture is a big part of that. If you eat, you're involved in agriculture. And I'm delighted that we're putting such a highlight on this sector of the economy and what's so important to so many people around the world. I don't know if Stephen Mayfield is here. I didn't like the way that he substituted tar for oil sands. We joked about that a little bit. But I found when he delineated, and last night I heard the need for a champion in terms of getting more research, he really laid it out very nicely with respect to the amount of activity that's involved in food production and agriculture as a component of total economic activity in the world. So we may have to bring up a guy from San Diego to be our champion. This panel will now introduce and be very forthright in describing their research. They've each got 3 minutes to talk about the future of food. You know, food is now morphing from being caloric intake for the world to producing fuels, and there are unknown boundaries yet to be pushed back. And so we have asked each of them to talk about the problem that their research addresses, the genomics solution they are developing through their research, the value proposition of their genomic solution over the current state of affairs or status quo, and the hurdles that stand in the way. And at the end, I think I really want to focus on the hurdles, because I've referred to funding, we've talked about pure research, the infrastructure that's required for research, so I really look forward to addressing what might be the hurdles to move forward. So I get to start with a colleague of mine at Livestock Gentech and a professor at the U of A, Graham Plastow. Graham comes from the UK, stole our bank governor, for heaven's sakes, so I hope we have the money to do something here now that he's left us. But Graham, would you start, please? And the ladies are keeping score. 3 minutes. We're going to be very strict.
Oh dear, I better stick to my script then. What a fantastic event it's been. It's almost nothing left to say, you could argue, if we didn't have our scripts. And I'm reminded with all of the fantastic talks of one of my favorite laws, and it's a law of Arthur C. Clarke, which is any sufficiently advanced technology is indistinguishable from magic. And I think we've heard some fantastic stories and some fantastic illustrations of where genomics is close to magic in many respects, and we get to use that. And this session is feeding the future, and I think the important thing, the problem is doubling food production in the next 40 years, and doing that, and with this here you can see with less land, water and energy, and as we've heard, the impact of climate change, and retaining our social license to operate. I think those are the key things. And as we move, from the livestock perspective, as we move to 2050, we're going to see 3 billion people entering the middle classes, and in fact, as was talked about this morning, wanting to eat in different ways. And the demand for animal protein is going to increase in this next period of time. So that's the challenge for us, and I think as that happens, then also people's expectations for how livestock are produced will change. So that social license part comes in. So as we've heard, the genomics can be a very important part of improving traits that are difficult and expensive to measure, even using the fantastic tools that we've used over the last 50 years to breed animals and increase production. Things like animal health, the health of animals, the welfare of animals, very difficult to make improvements to. And so the project that I'm working on, leading with a big team of people funded by Genome Canada and other partners, is actually looking at variation in susceptibility to two of the most important viruses in pig production. And they have a huge impact, probably something like $1 million a year in North America. The high mortality from these viruses, but also a big impact on growth and morbidity because those sick animals have to recover, and that impacts the efficiency of production. So, and in the pig picture, some of you may be able to see these were all the same weight at the beginning of the experiment, and you can see variation in size. And amazingly, although all of these pigs are producing virus, some of them are still growing at the same rate as their uninfected controls. And we have the basis to go in and find the regions that are explaining this variation. So hurdles, very quickly, as my time's run out, I think it's a case of if we're going to select for improved health, what are the trade-offs? What are the trade-offs in terms of the other traits that we're interested in producing? And then I think those are the answers we have to have if we're going to be able to take this technology to producers and to get, as I mentioned before, that social license to keep the production. Those are the sort of questions that we need to answer.
Okay, thank you, Graham. We will now move to Willie. I won't introduce you. Simon Fraser, salmon specialist. Go ahead.
Okay. So what you see up there is actually our discovery strategy for the salmon sequencing genome project. And what we see, starting from, I guess, this side over to there, are the resources that we've been producing, and then how will these be implemented into breeding programs primarily. So we'll be looking at more markers. We have SNPs, for example, array tools, vaccine strategies. Why are we doing this? Well, capture fisheries have reached a plateau. And if we're going to feed the world with healthy salmon, which is what we want to do, then it'll have to come from aquaculture. But we need a social license here. People want to eat salmon. They don't just want to eat fish protein. They want to eat delicious fish protein, and delicious fish protein with added nutrients like omega-3 fatty acids and the like. However, they don't want to have any impact on the wild fishery of any wild fisheries, whether it be salmon or be anchovies or other fish like that. And they don't want to have any impact on the environment. So we have to try and balance these. I mentioned we have to try and make salmon more environmentally friendly by eating plant protein rather than fish meal. Not that many of you have probably eaten fish meal as a staple in your diet. Breeding programs are in effect in some places. Norway has got a national breeding program. Some companies, such as in Tasmania, Saltas, have their breeding program. But there's no real big coordinated effort in the likes of Canada, for example. And our big hurdle is actually persuading the fish companies, the aquaculture companies, that using marker-assisted selection and the like is actually cost-effective. There is now an IPN haplotype, which means that the IPN is infectious pancreatic necrosis virus, and that is being essentially wiped out through breeding. That's a great proof of concept, and we hope to will develop more and that all of you will enjoy even more salmon in your diet.
Okay, thank you very much, and right on time, Willie. Very good. Now, we have quite a contingent from Saskatoon on this panel, in fact, 3 individuals. I'll start with the first of the 3, Fuzi Bakawi, who is the National Research Council Interim Director based out of Saskatoon, and he heads up the wheat flagship project, so I'm sure you can tell us much more about your plant breeding background, your initiatives, and you have 3 minutes.
Thank you. You've all heard the talk by Peter Langridge showing the importance of wheat in the world as being one of the most important crops in terms of protein intakes and production. Within Canada, wheat is crop number 1 in terms of acreage. It's important in terms of export. While Canada is ranked number 6 in terms of production, is in the top 3 in terms of export of wheat. To give you an example, Saskatchewan export approximately 10% of the overall wheat in the world. In terms of contribution to the economy, it's an $11 billion economy with production and related industries. So it's an important crop for Canada. The problem, I think that's also was highlighted by Peter Langridge. We need to improve genetic gain if we want to meet the demand in food. The rate, the genetic gain right now is not sufficient. It has to be improved by at least 60%. And some of the other challenges and the problems that we'll be facing is, one of them is climate change. We need to develop varieties that can be adapted to extremes, extreme cold, extreme heat, drought, and also adapt to resistance to disease and insects. Insects and disease evolve, so you have to keep producing new varieties. And reduce input of fertilizer, water, for example. Those cost money, they affect the environment. We have to reduce those while keeping the yield at a higher level. What are the solutions? The slides are prepared by my colleague, Curtis Pozniak, who leads the Genome Canada project, is trying to address some of these issues. And basically, we need to integrate genomics to breeding. Genomics provide several solutions. Some of the technologies and the tools are shown there. We will be able to discover markers, doing genomic selections. We'll be able to link traits to genes and being able to use that information to breed new varieties. We will combine those tools with others, such as double haploidy, to improve the yield for the benefit of the farmers. What are the hurdles? Again, Peter highlighted this, the complexity of the wheat genome. It's 17 billion bases, 5 times larger than the human genome, 40 times larger than the rice genome. With such complexity, we need the adequate tools, such as bioinformatics, to be able to process and generate all that data. This is, at the same time, a challenge, but an opportunity. I mean, a few years ago, it was unthinkable that we could sequence the wheat genome, but now we have the tools. We just have to develop them and apply them. And another hurdle is we have to be able to translate the research into breeding and basically integration of the breeding program. And this project also is a very good example. It involves biologists and genomics people at the same time as breeders. There are several breeders from Ag Canada and University of Saskatchewan involved in this project. And the last point I would like to make is that's really important is partnerships. This project, as you can see, involves several public, private, and international collaborators. We have to build on that. This project is 3 years. Breeding wheat takes 10 to 14 years. So we have to plan long term. And through the program that John McDougall mentions, that I've been working with my colleagues and also with Ag Canada, the University of Saskatchewan, the province of Saskatchewan, we developed this 11-year program to try to meet those challenges. But we can't do it alone. We have to involve industry and other public institutions and international collaborators.
Okay, thank you. Important message there with partnering. Now, Isobel Parkin, I'm going to have just a brief bit of fun with her, because if you go to your program, there's no picture. And randomly, just by accident, I sat by her the very first day. I introduced myself. She was kind of shy. I said, she said, I'm on your panel. And I said, well, I didn't see the picture. I didn't relate. And she said, yeah, I saw you had your picture. That must have been taken quite a while ago. So that was a bad start for me, but I still like her. She's from Saskatchewan. She's a plant geneticist, genome research. She's with AAFC, Agriculture, Agri-Food Canada. Isobel, come on. I'm never letting you forget that.
I know. I realize that. Okay, I apologize again. Yeah, so yes, I'm with Agriculture and Agri-Food Canada, and not that I'm trying to compete, but I work on canola, which is it's the second crop, I guess, in the prairies compared to wheat, but it's economically probably more viable. It has a $15 billion industry, and so it has a lot of value to Canada. But again, in comparison to wheat and salmon, we have the same problem with a very complex genome. It was formed from the fusion of two smaller genomes, and they themselves are actually hexaploids. So any trait that we work on, even simple traits, tend to be controlled by multiple genes, which means that anything we do is quite complex. And beyond that, most of the traits that people are interested in improving now, which is yield and then response to abiotic stresses, are themselves controlled by multiple metabolic pathways. And so we have a complex problem, and genomics offers the ability to look at a genome-wide solution. And so that's why we've turned to genomics. So we were fortunate to be able to develop a public-private consortium, which has been used to sequence the canola genome and its progenitor genomes. And we've already been using those sequences to develop a genome-wide marker set, which is highly valuable, as a number of the talks have mentioned, for breeding technologies and for the potential for genome selection in this crop. So we're now using this tool to not only sort of associate positive alleles for particular traits with regions of the genome, but also to start capturing diversity. And that, again, has been mentioned in a number of talks. And breeding has been excellent in selecting for particular traits, but it's also inadvertently created genetic bottlenecks because they've only selected for particular regions of the genome. So now we want to try and reverse that trend by bringing back some of the diversity that has been lost. So one of the hurdles, I guess, in this problem is that although it's very easy to capture sequence diversity, so with the massive changes in sequence technologies, we can sequence thousands of lines. But actually being able to utilize that diversity within our programs is a much more difficult proposition. I'm off. Phenotyping is one of the things that we're going to face as a big problem, and being able to develop sort of high throughput, robust systems for phenotyping large numbers of lines and identifying positive traits within those lines. And being able to develop high throughput phenotyping systems that actually could replicate field conditions, which was mentioned earlier by Peter Langridge, which is another problem that we face. But we believe that the genomics tools are going to give us a step ahead in being able to sort of facilitate and move more quickly to crops that will respond better to current needs and also current environments. And in Nebraska is not only the food crops we're working on, but we have related species that are now being developed for platforms for bioproducts and for biofuels. So we have an interesting species to work in, but also we now have the tools to be able to move more quickly.
Okay, thank you, Isobel. Very well done. Now we have a representative from our regulator, Canadian Food Inspection Agency, and Burton, I'm sorry I hadn't met you earlier, but Burton Blais, who is head of Research and Development Section for the Ottawa Carling Lab here. We look forward to your input on food safety and regulation.
Thank you very much. I'll be giving, I think, a slightly different outlook on genomics than what I've been hearing the last 2 days. I am primarily a food microbiology researcher, and the agency that I work for has a food inspection approach which is predicated on a risk-based approach to protecting Canadians from preventable illness. And what that means is that we seek to identify and control the entry points for hazards in the food production continuum. So that's from gate to plate. And part of that involves a program of inspection where there is a strong laboratory testing component, for example, detecting hazards such as food pathogens. And in the risk-based approach to food inspection, it's especially important to have methodologies that are not only able to give rapid results so that you have a short turnaround time on results to have very timely interventions, but also very informative results so that we can determine what is the nature of the hazard that's present, what is the extent of risk to public health that is connected with the laboratory result. We see genomics as having a lot of potential to, of course, expose the blueprint for microorganisms to help us understand what it is that makes them tick as pathogens. You know, what are the salient characteristics such as virulence factors and marker genes that would enable us to identify them definitively in food samples and maybe characterize or do risk profiling to determine the extent of risk to human health and therefore to have the best possible regulatory interventions in consequence so that we can best safeguard public health. And so genomics offers a great potential in this area, and there are actually some real-world examples where genomics has come to play in some fairly notorious cases of foodborne illness outbreaks in Canada. If you think of the maple leaf outbreak of 2008 that involved Listeria monocytogenes, our colleagues at the National Microbiology Laboratory at PHAC were able to sequence the causative agent from clinical samples within a day. And so real-time sequencing or near real-time sequencing, a whole genome sequence of pathogens in the context of an outbreak investigation is becoming a real possibility. Where we're having barriers right now is how to interpret that blueprint. How do you mine that data to identify the types of characteristics that would enable us to make a proper risk assessment? Very importantly, how do we find molecular signatures, molecular subtyping, characteristics that we can use in traceback investigation so that our labs in the CFIA side of the equation that have to do a lot of food testing when there is an outbreak situation can do the best job in terms of focusing in on, zeroing in on the problem. And the major holdback there is a need for more investment and development in bioinformatics tools and computing infrastructures, and certainly in our domain.
Okay, well, thank you, Burton. Very important work, and it's timely to, some of you may know, but many of you may not, that there's a funding consortium that is being put together by AI Bio from Alberta, Genome Alberta, ALMA, Genome Canada, and the province of Ontario in developing a rapid detection test for pathogenic E. coli with the specificity and sensitivity that we need. Very important for consumers, the industry, and international trade and market access. So genomics is going to play a fantastic role, and maybe I'll get you to comment a bit about that later. Our last Saskatoon panelist, Peter Phillips. Everybody needs an economist, right? You got to follow the money. You got to know the value. Where do you get your return, best bang for the buck? Peter was kind enough to bring his mother, a tremendous young lady here. She looks as young as he does, so that was nice to see. Not inferring that you're older, but your hair is grayer than mine, that's for sure. Peter talks about the economic impact of agriculture and ag biotech. So, Peter, take it away, please.
Thank you. I don't know whether anybody has caught the qualitative difference between yesterday and today. Yesterday was about fascinating science, but there wasn't much connection to the market and to the users except in an occasional comment. Every one of the panelists who've talked today have had a primary user as part of their teams and their networks. So one of the things that's different is that we are a big player in most of the fields we're talking about here. In Canada, we have comparative advantage. We're a global player, not just a niche player in these. And what we're finding is that we have a pathway to market in the agri-food and in the energy and in the forestry sectors, because we have those industries located here and they're able to take up and use it. The challenge is coordinating that. We have an alphabet soup of institutions who are making the science, building the science structure, and then gestating this science and trying to move it into the marketplace. And the challenge is that we have systems that are working in some areas, You know, we saw the graph a few minutes ago of corn versus wheat. One of the challenges is that wheat hasn't been as well organized as the private corn industry. Soy, similarly, has got a head of steam. Some of the other privately driven crops where the public sector really is a partner but not a driver are well organized, but there are parts that are critical to not only Canada but to the international food system that are not particularly well organized. So the challenge is organizing nationally, which is what some of that alphabet soup you see here and you've seen on these slides, but compounded by 30 to 50 times. Because not only do you have to satisfy domestic systems, you have to then translate this technology through the trade and science system into other applications and uses. So the GELS world is about trying to overcome some of those barriers. There's research design and research management tools, partnerships, international consortia, which we're learning by doing in many cases, and we're actually finding better practices, maybe not best practices. Second area is in the area of regulation. We heard a little bit about food safety, but the regulatory system, not only domestically but internationally, is not complete. Many of the things we heard about on the posters that we heard about yesterday and today, we don't have we have the risk analysis framework in place, but we don't have the operational guidelines and science and people to actually adjudicate and decide whether the technology should enter the market or not. So there's some major gaps in many of these areas. So the challenge then is to find exemplar institutional systems that can then be replicated across the agri-food and the industrial sector. Now the pitch really is that if you do ROIs on technological change, agriculture, bar none, has very high ROIs. The biggest ROIs are in institutional change, getting it right, because not only do you affect individual technologies, you affect the whole supply chain. You optimize the science that we've been talking about here today.